Welcome you all to the video lecture on the antibiotics penicillin. In the course Microbial Biochemistry, myself, Dr. S. Kadiravan, Assistant Professor in Biochemistry. In this video, we are going to see about antibiotics, discovery of penicillin, mechanism of action, production and uses. Antibiotic. The antibiotics are secondary metabolites or biochemicals secreted by microorganisms which in low concentration inhibit the growth or kill other microorganisms. That is, the antibiotics are antimicrobial agents of microbial origin. So antibiotics is not a new term for us, and we are using antibiotics in our day-to-day life. Whenever we are infected with some infection, we are given the antibiotic. So these antibiotics are the origin of microorganisms. So the antimicrobial antibiotics refers to the biochemical substances that is synthesized by the microorganisms. Although some 6,000 antibiotics are known to exist, only about 100 are marketed. Antibiotic medications are widely used in the treatment and prevention of such infections. They may either kill or inhibit the growth of the bacteria. So in general, antibiotics are the biochemical compounds that is synthesized by the microorganisms and they are released out of the cells, so that those antibiotics will act against some other bacterial species. So that mechanism is encountered and that mechanism is exploited in the usage of antibiotics. So as we saw in the previous slide, there are number of antibiotics that is used for different purposes. So each and every antibiotic has its own mode of action in order to prevent or in order to cure a disease. So this picture is the photograph of Sir Alexander Fleming, who discovered the penicillin, one of the landmarks in the history of the discovery of antibiotics. For penicillin, we are going to discuss about penicillin. Penicillin is a group of compounds having common basic nucleus 6-amino penicillinic acid, that is 6-APA. Penicillin are of two different types. One is natural and another one is semi-synthetic penicillin. And you can see this is the structure of a penicillin, which contains acyl side chain, beta-lactam ring and thiazolidine ring. So these three components together form the penicillin structure. Okay. Coming to the discovery of penicillin, how the penicillin was discovered. Let us see here is. So in 1928, Scottish biologist Alexander Fleming discovered that the Staphylococcus culture had been mistakenly left grown in open was contaminated with a mold which had destroyed the bacteria. After isolating a sample and testing it, he found that it belonged to Penicillium family. Later, the mold was classified as Penicillium notatum. So it is a surprise discovery. This penicillin discovery is often referred to as the accidental discovery or a surprise discovery because it is not intended for the discovery of penicillin. So while doing a set of experiments in a laboratory, Alexander Fleming accidentally found out the penicillin. At first, it was difficult to convince people about its potential uses, but later, in 1939, using Fleming's work, two medical researchers, Howard Florey and Ernest Chain, managed to purify the penicillin in a powdered form. In 1941, they successfully treated a human with this penicillin antibiotic. And in 1943, they produced penicillin on a large scale, and this produced penicillin was used for that treatment during the World War II, which had a great impact on the soldiers. Okay. This slide shows you the structure of a penicillin or the morphology of a penicillin. So it has, as it is a fungus, basically penicillium is a fungus, and the compound that is isolated from penicillium is a penicillin, and you can see the hyphae making up the mycelium, and the structure containing the spores. So this is a typical morphology of any kind of a fungi. Okay. So this is a morphology of a Penicillium fungus. And you can see the structure. This is a chemical structure of a penicillin, and you can see by altering the different groups on the penicillin, we get different drugs as penicillium G, penicillium V, penicillin V, ampicillin, amoxicillin, and methicillin. These are all the different drugs that are derived from penicillin. Coming to mechanism of action, it inhibits the synthesis of peptidoglycan in cell walls. So hope we all know that peptidoglycan is a component of the bacterial cell walls. So the antibiotics or the substances synthesized by the bacteria in order to kill the other bacterial species, a competent bacterial species. So in order to kill a bacterial species, the cell wall of the bacteria has to be damaged. If a cell wall has to be damaged, obviously it is the peptidoglycan that has to be damaged. So this penicillin can inhibit the synthesis of the peptidoglycan in the cell walls. Beta-lactam of penicillin binds to the enzyme transpeptidase that is used in the formation of a peptidoglycan cross-linking. Okay. So the enzyme is inhibited, thus inability to form the cross-linking. If the cross-linking cannot be done, it prevents the formation of the peptidoglycan cell wall. Okay. So therefore the cell is weakened, causing osmotic imbalance in the cell. This leads to the cell death. So this is how penicillin acts on a bacteria. So by degrading the cell wall of the bacteria or by preventing the formation of the cell wall of the bacteria may be weakened, and ultimately the bacteria can be removed from the position of the inhabitant. Okay. Coming to microorganisms which can able to produce penicillin, right? Out of the various species of fungus, Penicillium, mainly two species are used in the fermentation. These are Penicillium notatum and Penicillium chrysogenum. Even from these two, Penicillium chrysogenum is high yielding strain and therefore most widely used as production strain. The production strain is improved by mutation with the help of X-rays or any other agents to give high yield. After strain improvement, the production strain should be carefully maintained because Penicillium chrysogenum is genetically unstable, because it can even lead to some mutations. Okay. So this slide depicts you the flowchart of the biosynthesis of penicillin. So with the investment of L-alpha aminoadipic acid and with the presence of L-cysteine and L-valine, you can see several cyclization steps are carried out. Ultimately, we can able to produce the benzyl penicillin. So that benzyl penicillin is the penicillin actually that is used as an antibiotic. Okay. So what are the bioparameters that has to be maintained during the production of a penicillin in an industrial scale? pH nearly 6.5 should be maintained. Temperature, it's 26 degrees Celsius to 28 degrees Celsius. Aeration, a continuous stream of sterilized air is pumped into it. And agitation, have baffles which allow constant agitation. And fermentation broth consists all the necessary elements required for the proliferation of the microorganisms. Coming to nutrient sources, lactose acts as a very satisfactory carbon compound if it is used in a concentration of 6%. Other carbohydrates like glucose, sucrose may be used, but it has to provide with a slow feeding rate. Ammonium salts such as ammonium sulfate, ammonium acetate, ammonium lactate, or ammonia gas are used as nitrogen source. Mineral elements include phosphorus, sulfur, magnesium, zinc, iron, and copper, which is generally added in the form of a water-soluble salts. Depending upon the precursors added, the type of penicillin going to be produced can be changed. As we all know, there are several penicillins that are available in the market now, which can be produced by altering the nutrient concentrations present in the bioreactor. And this slide depicts the flowchart of the large-scale production of penicillin. You can see with the help of the culture, that is a Penicillium chrysogenum, we can able to produce the penicillin at a large scale. So with the help of the culture, so the stored culture has to be transferred to a shaker flask where the mycelium will be developed. And from that transfer vessel, from that shaker flask, it is transferred to a transfer vessel where it is further multiplied. And from the transfer vessel, it is added to the seed tank. So seed tank is a tank or a reservoir within which the microorganisms will proliferate in a manner so that acts as a seed culture for the large scale production of a particular compound. And then it is followed by the fermentation tank where actually the microorganisms proliferate in the presence of all the nutrient and the environmental conditions. And followed the fermentation tank is the rotary filter. So once the product is formed, that is the penicillin is formed, it has to be filtered from all the other suspensions present in the growth medium, and followed with the first extractor, followed with the purification column, second extractor, third extractor, vacuum still, and then ceramic filter, crystal wash, ceramic filter, and vacuum dryer. At last, crystallic potassium penicillin G is extracted as an antibiotic. Okay. Coming to recovery, the recovery of penicillin is carried out in 3 successive stages. That is, one is removal of mycelium, counter-current solvent extraction of penicillin, and treatment of crude extracts. So these are the 3 stages from which we can able to extract the penicillin. So one is removal of mycelium. Removal of mycelium means, so mycelium is known as the vegetative part of the fungus, and from the mycelium only the penicillin will be extracted. And by extracting the penicillin alone, we need not need the mycelium. So it has to be removed, and counter-current solvent extraction of penicillin. So as there are, not only the penicillin will be synthesized during the fermentation process, several other compounds will also be synthesized during the fermentation process, but we are concerned only about penicillin. So we have to remove all the other compounds from the extraction. So that is, for that process, counter-current solvent extraction is utilized, and the treatment of crude extracts. So there will be several solvents, several solutes that would have been dissolved in these solvents. So we have to treat the crude extracts in order to get the purified penicillin. So that is also done by several methods. Coming to uses of penicillin, most of the penicillins are active against gram-positive bacteria, in which they inhibit the cell wall synthesis, leading to the death of the bacteria. So penicillin is a compound that is produced by a fungus which can act against the action of a bacteria. Used therapeutically in the treatment of infectious diseases of humans caused by the gram-positive bacteria. So this is about the major uses of penicillin. So since the discovery of penicillin have created a great impact in the treatment of diseases that is caused by the bacterial poisoning, bacterial diseases. So this slide shows an overview or summary about the process of penicillin production. You can see with the help of the penicillium mold that acts as a source for the production of penicillin, and with the help of the fermentation tank we can able to produce the penicillin, and we can able to separate the penicillin from all the other molecules, and the purified penicillin is used as an antibiotic for the human consumption. So you can see this is the penicillin that is available in the market. So you can see penicillin G potassium for an injection is available. It is available as an injection, and also penicillin G sodium for injection, that is also used. So in different forms, penicillin is present as a sodium salt, or it is present as a potassium salt. Penicillin is always a penicillin. So in order to maintain the pharmaceutical preparation, in order to maintain the efficacy, in order to ensure the stability of the product, some forms will be prepared. So these are all some of the forms in which penicillin is available. Okay. In a nutshell, we have learned about what is antibiotics, and we saw about the discovery of penicillin, mechanism of action of penicillin, and production of penicillin, and uses of penicillin. Thank you.